Here's a question almost no one asks themselves. If you want to change, why does your body resist it so aggressively? You set goals, you feel motivated, you start, and then, a week later, a month later, you find yourself back in the same place, the same thoughts, the same reactions, the same results. Most people call it laziness or weak willpower. But here's what you need to understand. This is not psychology, it's biochemistry. Your body is literally chemically addicted to the version of you from the past. And every time you try to become someone new, your cells, like real addicts, demand the old dose of the old hormones, the old emotions, the old identity. Joe Dispenza, a neuroscientist who studied hundreds of cases of spontaneous healing, calls this addiction to the past self. In a few minutes, you'll get the exact biochemical algorithm to break out of this loop. But first, we need to look inside, the chemical factory that's running your life.
Deep in the center of your brain, there's a structure about the size of a large grape. It's the hypothalamus, and it literally decides who you are. Here's how it works. Every time you experience an emotion, fear, joy, anxiety, excitement, the hypothalamus instantly translates that emotion into a chemical signal. It produces special molecules, neuropeptides. These are small chains of amino acids, a kind of chemical language that the brain sends to the body. These neuropeptides reach the pituitary gland, the master endocrine gland, and trigger a cascade. Hormones are released into the bloodstream: cortisol, adrenaline, oxytocin, dopamine. Each emotion equals a specific hormonal cocktail. Each hormonal cocktail equals a physical state of the body. Each physical state equals behavior. A metaphor Dispenza often uses. Imagine your hypothalamus as a barista in the coffee shop of your nervous system. Every day, it prepares the same order because that's what you've been ordering thousands of times. Fear, anxiety, dissatisfaction, that's your usual latte. And your body has learned to expect it. But here's where it gets strange. The hypothalamus does not distinguish between what's real and what's imagined.
In 1995, neuroscientists conducted an experiment that changed how we understand the brain. Two groups of pianists. The first group physically practiced the same passage 2 hours a day for 5 days. The second group only imagined playing mentally without touching the keys. MRI scans after the experiment showed almost identical changes in the motor cortex in both groups. This is not a metaphor. This is neurophysiology. Your hypothalamus has no sensory organ to check, is this actually happening, or am I just thinking about it? For it, the experience and the memory of the experience are biochemically equivalent. This means the following. If every morning you wake up and automatically start running thoughts about money, work, the future, your body begins the day in a chemical state of real threat, cortisol, adrenaline, a fight or flight state from the morning without a real enemy. And in this state, you try to build a business, raise children, create relationships. Most people make the same mistake here. They think it's enough to just think positively. But without changing the chemistry, it's just words layered on top of a hormonal storm.
Now, the most important part, and the most uncomfortable. The pattern thought-hormone is not just a habit in a psychological sense. It is a physical structure. Every time you experience the same emotion, anxiety when thinking about money, irritation when thinking about a specific person, hopelessness when thinking about your future, the neurons that fire together literally wire together. Hebb's rule, one of the fundamental principles of neuroscience. Neurons that fire together wire together. After thousands of repetitions, the same thought triggers the same emotion automatically, without your participation, like a reflex. But that's not all. The receptors of your cells, all 37 trillion cells in your body, gradually adapt to your standard hormonal state. They increase the number of receptors for the hormone that is most common. They reduce sensitivity to rare hormones. This means if you live in stress for years, your cells literally crave cortisol. The absence of anxiety starts to feel like discomfort, like something is wrong. Dispenza calls this cellular addiction, and you cannot break this addiction by decision alone. You have to change the chemistry.
In survival mode, your body does one thing brilliantly well. It mobilizes all resources to survive right now. The adrenal glands release cortisol and adrenaline. Blood is redirected from internal organs to the muscles. Vision narrows, thinking simplifies, only the immediate threat matters. Perfect for escaping a predator. Disastrous for building a business, writing a book, or creating relationships. But here's the paradox. Survival mode becomes addicted to itself. When cortisol is chronically high, the brain starts to treat it as the norm. The prefrontal cortex, the part of the brain responsible for long-term planning, creativity, empathy, innovation, literally shuts down, because in survival mode, long-term thinking is a luxury. Research shows chronic stress reduces the volume of the prefrontal cortex, physically reduces it. This means the longer a person stays in survival mode, the less of their brain they actually use to create their life. And here's the key question that leads to Dispenza's method. If the body is the subconscious, if it stores programs in its cells, how exactly do you reprogram it? In 30 seconds, you'll see how to change this hormonal state. This is where the real science of change begins. Neuroplasticity is the proven ability of the brain to form new neural connections throughout life. This is not a self-help metaphor. It is a literal, physical restructuring. But here is Dispenza's key insight, the one that changes everything. Since the hypothalamus does not distinguish between what is real and what is imagined, we can consciously create the hormonal states of the future in the present. If you can vividly and in detail experience in your mind the state of success, achievement, safety, love, your hypothalamus will begin to produce the neuropeptides of those emotions. The pituitary gland will release those hormones. Your cells will receive a new chemical signal. Dispenza calls this being in the future emotionally before it happens physically. This is not positive thinking. This is not affirmations in the mirror. This is controlled biochemical influence on your own hypothalamus. The difference is fundamental, and affirmation is a word in the mind. Emotional rehearsal is an experience in the body. One does not change chemistry, the other does. But there is a very specific algorithm here, and most people get it wrong at the very first step.
Step 1, the one most people skip, is called breaking the autopilot loop. Here's what happens for the average person every morning. The alarm goes off. Before even opening their eyes, the brain starts loading yesterday's state. Anxieties, unfinished tasks, fears, resentment. This is not weak willpower. It's a neurochemical reflex. Neural pathways, built over years, activate automatically. Just like you don't think about how to move your legs when you walk. The first step of Dispenza's method is to literally catch this moment. Technically, this is called metacognition, the ability to observe your own thinking. When you notice an automatic thought, it no longer fully controls you. You become the observer, not the content. At the level of neurobiology, this activates the medial prefrontal cortex, the very region suppressed by chronic stress. In practice, it looks like this. You wake up, and instead of reaching for your phone, you do one simple action. You ask yourself, who do I want to be today? Not what to do, but who to be. This question is a biochemical key, but only if it is followed by step 2.
Dispenza draws on research from the HeartMath Institute.
Here's what they found. When a person experiences chronic stress, the heart rhythm becomes chaotic, irregular, disordered. This chaotic signal travels through the vagus nerve to the brain and literally disrupts cortical function. When a person experiences states like gratitude, love, inspiration, the heart rhythm becomes coherent, smooth, sinusoidal, ordered. This coherent signal travels to the brain and synchronizes the entire neural system. This means emotion is initiated by the body, not just by thought. Step 2 of the algorithm is the breathing protocol of heart coherence. A simple technique. Inhale for 5 seconds. Exhale for 5 seconds, even without pauses, for 3 to 5 minutes. This is enough to activate the parasympathetic nervous system, reduce cortisol, and shift the heart rhythm into a coherent state. But there is one critical element without which this is just breathing. Neuroscientists at the HeartMath Institute found coherence reaches its peak when during breathing a person consciously focuses attention on the area of the heart and evokes a memory that generates a genuine feeling of gratitude. Not the word gratitude, but the actual experience. This is biochemical key number 2. The next step is the most powerful.
This is the most critical step, and this is where most people either stay superficial or stop entirely. After you've brought the body into coherence, cortisol reduced, prefrontal cortex active, nervous system in rest and recovery, you are ready for emotional rehearsal. Here is the exact algorithm. Step 3A. Define one specific state, not a goal, but a feeling. Not, I want to earn $10,000, but what will I feel when it happens? Freedom, confidence, calm, inspiration. This is a specific emotion, your target hormonal cocktail. Step 3B. Create a sensory-rich mental scene, not an abstract image, but an experience. What you see, hear, feel in your body, what you say, what others say, what the space around you looks like. The more detailed the scene, the more convincing the signal for the hypothalamus. Step 3C. Do not visualize from the outside. Enter it. Most people watch their visualization like a movie. That does not work. You have to be inside it, seeing everything from the first-person perspective. This is what switches the hypothalamus from this is imagination to this is happening.
One session changes nothing. And this is not a weakness of the method, it's biology. Here's why. When you build a new neural pathway, it's like clearing a trail through a dense forest. After the first pass, barely visible. After the 10th, a path. After the 100th, a road. Neuroscientists call this process myelination. Nerve fibers become coated with a white insulating layer, myelin, which increases signal transmission speed up to 100 times. In practice, the minimum period for a new emotional pattern to start feeling natural is 21 days of daily practice. For deeper restructuring of cellular receptors, Dispenza speaks of 63 days, 3 cycles of 21 days. Morning, immediately after waking, before the old program loads. Evening, before sleep, when the brain shifts into a highly plastic state, the alpha-theta boundary. Duration, 15 to 30 minutes. This is the minimum dose for biochemical change. But here's where it gets strange. After 3 to 4 weeks of practice, people begin to notice changes, not only in how they feel, but in their external circumstances. And here is why that happens from a neurobiological perspective.
In the brainstem, there is a small neural network called the reticular activating system, the RAS. Its function is to filter information. Every second, your sensory systems receive about 11 million bits of information. Conscious awareness can process only 40 to 50 bits. The RAS decides what reaches consciousness, and it makes that decision like this. It allows through whatever matches your dominant emotional states and beliefs. This explains a well-known phenomenon. You buy a red car, and suddenly you see red cars everywhere. They were always there. The RAS simply did not mark them as relevant. When you spend months practicing emotional states of confidence, possibility, abundance, your RAS begins to literally perceive a different reality. It notices different opportunities, hears different conversations, pays attention to different people. Neurobiologically, this is called attentional filter recalibration. Dispenza describes it as attraction, not mystical, but neurochemical. Your body, in a new chemical state, begins to behave differently, subtly but consistently. Different body language, different decisions, different words, and that produces different results.
Survival mode is the chemistry of the past. Cortisol, constant mobilization, anxiety, sleep disruption, immune suppression. Adrenaline, short bursts of energy, fight or flight reactions. Narrowed perception, short-term thinking. The body lives in the past because past experience equals threat prediction. Creation mode is the chemistry of the future. Oxytocin, trust, connection, openness, safety. Dopamine, anticipation, motivation, movement toward goals. Serotonin, stability, confidence, self-worth. Anandamide, the bliss molecule, creative flow, intuition. The difference between these states is not optimism or pessimism. It is literally two different biochemical organisms. One sees threats, where the other sees possibilities. One depletes resources, the other generates them. And switching between these modes is not a matter of willpower. It is a matter of conscious regulation of your own hormonal system through a specific protocol. The algorithm described here is not philosophy. It is applied neurochemistry. But there is one final obstacle, rarely spoken about.
Dispenza calls this the known self, a collection of all habitual thoughts, emotions, reactions, repeated often enough to become identity, your I. And here is the problem. When you begin practicing a new emotional state, the known self resists. Physically, literally, the body will send signals of discomfort, boredom, a sense of this is not real, this feels forced. Thoughts appear. This is not working. This is not for me. I'm just pretending. This is an abstinence response from cellular dependency. Cells are not receiving their familiar hormonal cocktail. They signal distress. Understanding this mechanism is critical. Most people at this stage interpret discomfort as a signal to stop. It is not. It is a signal that change has begun. Dispenza describes this as the death of the old personality. It is biochemically predictable. People who understand this process move through it. People who don't return to familiar discomfort, because familiar does not mean beneficial. Familiar means habitual. Claiming scientific consensus here is overstated. The real picture is narrower, more mixed, and less absolute. HeartMath Institute has published work on heart rate variability, emotional regulation, and stress reduction. Their findings generally support that paced breathing and positive emotional focus can shift autonomic balance toward parasympathetic dominance and improve HRV. The stronger evidence is for stress modulation, not for broad claims about reprogramming reality or direct causal reshaping of life outcomes.
Richard Davidson and the University of Wisconsin-Madison have produced robust neuroimaging research showing that meditation and mindfulness practices can alter brain function and, in some cases, cortical thickness or connectivity in regions involved in attention and emotion regulation. These effects are real, but they are gradual, moderate, and task-specific. Improved regulation, not personality replacement or instant biochemical state switching. Work associated with Ellen Langer, Harvard University, on mindfulness and counterclockwise style interventions suggests that psychological context and expectation can influence perceived well-being and some physiological markers.
However, many of the stronger claims about reversing aging or large physical changes are debated and not consistently replicated in full form. Epigenetics is also often overstated in popular discourse. It is true that gene expression can change in response to environment, stress, behavior, and learning. It is not true that short-term thoughts directly rewrite DNA or produce large-scale, rapid genetic reprogramming. Most epigenetic changes are slow, context-dependent, and constrained by biology. The accurate synthesis is simpler. Stress regulation, attention training, and emotional control can measurably shift nervous system activity, hormone levels, and brain network function. These changes can improve behavior consistency and decision-making. Over time, behavior change can alter life outcomes. The exaggerated version turns this into direct reality selection or deterministic biological identity switching. That step goes beyond current evidence.
Epigenetics does not support that level of deterministic control or rapid transformation. What is accurate? Bruce Lipton popularized the idea that environmental signals influence cells, and in general terms, this aligns with biology. Cells respond to chemical environments, and gene expression is regulated. Real epigenetics involves mechanisms like DNA methylation, histone modification, and chromatin remodeling. These mechanisms do regulate which genes are more or less active. They respond to factors such as long-term stress, inflammation, nutrition, sleep patterns, toxins, and developmental environment. Chronic stress is associated in research with changes in inflammatory signaling pathways, immune regulation, and stress response systems. But this is not a simple switch between bad genes on and good genes on. It is multi-layered regulation with trade-offs, thresholds, and long timelines.
The claim that emotional states like gratitude or love directly activate regeneration genes in a targeted, programmable way is not supported by mainstream molecular biology. Emotional states can influence physiology indirectly through autonomic and endocrine pathways, but gene expression changes are not that precise or intention-driven. Joe Dispenza frames this as you author your genes, but scientifically that is a metaphor. In biology, you influence gene expression patterns through behavior and environment over time, not through immediate conscious emotional control. The accurate version is, repeated psychological states can influence stress physiology. Stress physiology can influence signaling pathways. Over long periods, these signals can correlate with changes in gene expression patterns. These effects are probabilistic, partial, and context-dependent, not direct control of genetic output through emotion. The core correction: the body is responsive to environment and behavior, but it is not directly programmable by transient emotional states in the way described. Presented as a structured protocol, this aligns with known elements of cognitive and physiological regulation, but the mechanism needs strict boundary correction. Step 1. Autopilot interruption. Morning, before stimulation. This is attentional control. The moment of waking is a low interference state. Asking a self-referential question shifts activity toward executive networks, prefrontal cortex engagement. This does not reset identity, but it can interrupt habitual rumination loops. Step 2. Coherent breathing, 3 to 5 minutes. 5-second inhale, 5-second exhale produces paced respiration near 0.1 Hz, which is associated with increased heart rate variability and parasympathetic activation. Focused attention can further stabilize autonomic output. Effects are real in stress reduction and arousal regulation, not state creation beyond physiology. Step 3. Target affect labeling. Selecting a single emotional label, calm, confidence, is affect labeling. This recruits regulatory circuits and can reduce limbic reactivity. It does not define a future hormonal state, but it can bias current emotional processing. Step 4. Multisensory imagery. Vivid first-person imagery activates overlapping networks with perception and motor simulation, visual, auditory, somatosensory cortices. This strengthens neural encoding through repetition, especially when emotionally salient. Step 5. Sustained immersive imagery, 10 to 15 minutes.
Sustained internal simulation increases functional coupling between default mode network and emotional processing circuits. The effect is training-dependent. It is learning, not direct biological state switching. Step 6. Somatic anchoring. Pairing a mental state with a physical gesture is associative learning, conditioned response formation. The brain links internal state plus motor pattern. This can improve recall of a mental state under stress. Step 7. Evening repetition, pre-sleep. Pre-sleep periods increase memory consolidation and synaptic reactivation. Rehearsed content can be strengthened during sleep-related consolidation processes. This supports learning stabilization, not direct subconscious reprogramming. Time frame claims, 21-63 days. There is no fixed universal biological threshold for identity change or cellular reprogramming. Behavioral habit formation varies widely, often weeks to months depending on complexity, repetition frequency, and reinforcement context. Neuroplasticity is continuous, not cycle-based. Core correction. This protocol is best described as attention training, autonomic regulation, imagery-based learning, plus habit reinforcement, not direct control of hormones, genes, or external reality outcomes. These 3 errors can be translated into established learning and stress physiology without assuming the stronger claims about hormones, hypothalamic response to intention, or withdrawal from identity. Error 1. Imagery without affective engagement. Mental imagery alone produces weak and inconsistent activation. When imagery is paired with emotional arousal, there is stronger recruitment of limbic and sensory networks, which improves memory encoding and learning. The key mechanism is emotional salience, not hypothalamic response to images. Error 2. Practice under high stress. High cortisol states are associated with reduced prefrontal cortex efficiency, impaired working memory, and increased habitual responding. Practicing attention-based tasks under high physiological stress reduces performance quality. Down-regulating arousal first can improve cognitive control. The coherence before training sequence matches basic arousal regulation principles. Error 3. Expectation of rapid change and early dropout. Early discontinuation is a known feature of habit formation attempts. Initial discomfort often reflects cognitive load, attentional effort, and mismatch between intention and automatic behavior patterns. This is not biological withdrawal from a past self, but competition between established habits and newly trained responses. Persistence allows repetition-based consolidation of new behavioral patterns. Core correction. What is described here is a combination of emotion-memory coupling, stress modulation before cognitive task performance, habit extinction and replacement dynamics, not direct dismantling of neural identity structures or predictable biochemical resistance of cells to change. Let's summarize what you now understand. Your hypothalamus is a chemical factory that over years has produced the same product: stress neuropeptides, anxiety, survival mode chemistry. Your cells have adapted to this product. They expect it. They rely on it. This is not your fault. This is your biology. But biology is not destiny. Neuroplasticity, epigenetics, and heart coherence are not self-help concepts. They are peer-reviewed science that points to one conclusion: you can consciously change the chemistry of your body. Not through willpower, through protocol. 7 steps, 21 days, 15 to 30 minutes a day. This is not a high price for the ability to live in the chemistry of creation instead of the chemistry of survival. Start tomorrow morning, not with your phone, with the question, who do I want to be today? Give yourself 5 minutes of coherent breathing. Enter one sensory scene. Hold the emotion in the body for at least 3 minutes. This is the first neural trace, the first step toward a new biochemistry. Your body is not a prison. It is a laboratory, and you are its primary scientist.